We're pleased to have Kiniksa Pharmaceuticals here at the Wedbush PacGrow Healthcare Conference. With us today are both Sanj Patel and John Paolini, and actually the rest of the management team here too, but to introduce the story, at least at first, is Sanj Patel, CEO.
Thank you, David. Good afternoon. It's great to be here in New York City. As I mentioned, John Paolini, our Chief Medical Officer, and I will review the data today. Thanks to Wedbush for the invitation to present this morning, as well as David. Our mantra at Kiniksa is every second counts. So let's get started. Please note that we will be making forward-looking statements today that are subject to risks and uncertainties. A review of these risk factors are contained within this slide, as well as under the heading of risk factors in our SEC filings. Trying to get to the next slide. There you go. Kiniksa is focused on discovering, acquiring, developing, and commercializing therapies to help patients transform their lives. All of our programs are based on either strong biologic rationale or validated mechanisms. And they're all focused on rare diseases with unmet medical needs. And importantly, all of our programs also have the potential to be a pipeline within a molecule. We've got a brilliant team, and one that's very much focused on capital allocation. And in fact, that team has executed on all of our communicated timelines, actually since starting the company. Our development strategy focuses on modulating central control nodes of the immune system. And in fact, the autoinflammatory and autoimmune markets have grown significantly in recent years, and there remains significant unmet medical need, as well as potentially attractive commercial opportunities. And looking at this slide, you can see that we've amassed a portfolio of autoinflammatory and autoimmune product candidates. And they range from a spectrum of diseases, from autoinflammatory through to autoimmunity, as well as from the innate to the adaptive immune system. This is our pipeline. We're enrolling in a single pivotal phase 3 study with Rolana-cept, and that's in recurrent pericarditis. This is a debilitating, painful autoinflammatory cardiovascular disease. We're also enrolling in a global phase 2 study with Mavar-illuma, and that's in giant cell arteritis, or GCA. And this is also a debilitating autoinflammatory disease that involves inflammation of the medium to large blood vessels, also potentially resulting in permanent blindness. We're enrolling in a phase 2A study in prurigo nodularis. This is also a chronic, debilitating autoinflammatory skin condition. And on top of that, we're targeting multiple indications that are characterized by chronic pruritus in a phase 2A exploratory study. KPL-404 and 045 are our preclinical programs, and importantly, we plan to file an IND for KPL-404 this year, making potentially now 4 clinical stage programs for Kiniksa. As I mentioned, all of our programs are based on either validated mechanisms or strong biologic rationale. And looking through the mechanisms and the rationale, you'll see that IL-1 alpha and IL-1 beta are cytokines that have shown to play a key role in inflammatory diseases. And in fact, Rolana-cept blocks the signaling of both IL-1 alpha and IL-1 beta. For the next program, GM-CSF is a key growth factor and cytokine that's involved in autoinflammation and autoimmunity, and Mavar-illuma blocks the signaling of GM-CSFR. And IL-31 and oncostatin M are also 2 key cytokines that are implicated in inflammation, fibrosis, and pruritus. And KPL-716 is designed to block both of those cytokines. So all our programs target unmet needs and have potentially attractive commercial opportunities in front of them. In fact, for recurrent pericarditis, there are no current FDA-approved therapies. And based on the analysis of multiple claims data, we see that the approximate U.S. prevalence is 40,000 patients that are seeking medical treatment. In terms of giant cell arteritis, there's only one approved product, and that's as an adjunct to steroid therapy. And also that particular program, we don't believe, addresses the underlying cause of the disease or the inflammation. whereas Mavar-illuma would be a first and best in class potentially. And the estimated U.S. prevalence for giant cell arteritis is between 75 and 150,000 patients in the U.S. And there's no approved FDA therapy for prurigo nodularis, again, a chronic, devastating autoinflammatory skin condition. And the estimated U.S. prevalence here is approximately 300,000 patients. So as Rolana-cept is the closest to commercialization for Kiniksa, I'm going to walk you through the high burden of disease for these patients that are suffering from this debilitating and painful disease. The highest rate of incidence of recurrences occurs in the refractory and steroid-dependent patients. In fact, 30 to 40% of patients have more than 2 recurrences, painful recurrences, within a year. And it's also that population that has the highest amount of cardiac complications, such as cardiac tamponade and constructive pericarditis. In fact, a lot of patients actually resort to opioid use to try and control their pain. And there's also a significant amount of anxiety and depression associated with the unpredictability of the disease and the severity of the disease. Next slide. There you go. So within the 40,000 patients that are seeking medical treatment for recurrent pericarditis, we believe there's around 14,000 patients that could be addressed with Rolana-cept. And our plan is to initially focus on the refractory patients. That's around approximately 3,000 patients within that 14,000 approximate addressable number. And then with positive physician experience and payer access, we plan to ultimately help treat the poorly controlled patients. That's a larger bucket, around 6,000 within the 14,000. And ultimately aim to become a steroid-sparing option. Our plan to reach our target audience is to have around 30 to 50 sales reps, cardiology specialty sales force. We're already ramping up our commercial plans. Upon approval, Rolana-cept would be the only FDA-approved therapy for recurrent pericarditis. So we're going to walk you through a number of the milestones before John digs into the pipeline programs a bit deeper, but we have a number of near-term events coming up this year, starting with the full phase 2 data from the Rolana-cept study, and that'll be coming in the second half of this year. In the first half of next year, we'll have data from the phase 2A study with prurigo nodularis. Just yesterday, we released some positive data, some rapid and strong reductions in pruritus from our repeated single-dose study. And we'll have that phase 2A prurigo nodularis first half. In addition to that, during 2020, we'll have a number of data cohort readouts from our chronic pruritus indications. In the second half of next year, we have data from the phase 3 pivotal Rolana-cept study in recurrent pericarditis. And on top of that, we'll have the global phase 2 study data from Mavar-illuma in giant cell arteritis. So a number of milestones there. I'll walk you through some of those again in a few seconds. But with that, I'll hand it over to John to go through the programs. Okay.
Thank you, Sanj, and good afternoon, everyone. So I'd like to walk you through, now it advances. We'll start with the KPL-716 data, since that's the data release that we had yesterday, and then we'll walk through the rest of the programs. So by way of reminder, KPL-716 is a monoclonal antibody which is designed to inhibit 2 key cytokine pathways simultaneously, IL-31 and oncostatin M, and it does that by targeting a single epitope, the shared receptor subunit OSMR beta. And the first indication that we are pursuing is prurigo nodularis, which, as Sanj mentioned, is a chronic skin condition with terrible pruritic nodules. By way of reminder, these are the data from the single ascending dose study that we presented at EADV last year. And you'll remember that we studied atopic dermatitis as a proxy for IL-31-driven pruritic diseases. Now, KPL-716 at the 7.5 milligram per kilo IV dose provided evidence of target engagement in terms of reduction in pruritus in this study of 20 subjects, and that was over a 28-day monotherapy period. Now, the reduction in pruritus, as you see here on the left, occurred within the first week of study drug and was persistent throughout at least the 4 weeks that were observed there, and perhaps out into about 6 weeks, which is consistent with the prolonged elimination pharmacokinetics of KPL-716 in the study at that dose. And on the right, what you see is what we saw as an encouraging early signal of efficacy in terms of reduction in atopic dermatitis disease markers, as measured by EASI. So then we proceeded forward with this repeated single-dose study, which is a randomized, double-blind, placebo-controlled trial in approximately 43 individuals. And the purpose of the study was to evaluate safety and efficacy of KPL-716 over much longer exposure periods. And we administered the drug as 360 milligrams subcutaneously every week for 12 doses. In order to replicate and extend the exposures that we saw with the 7.5 milligram per kilo IV dose. And what we've presented is an interim analysis of all subjects having completed the 12-week treatment period. And what you see is that there are no serious adverse events, and overall well tolerated, but there were more atopic dermatitis flares in the KPL-716 arm compared to the placebo arm. All of these atopic dermatitis flares were managed by the investigator successfully with topical corticosteroids. Okay. These are the data with regard to worst itch NRS in the interim period through 12 weeks of treatment. And what you see on the right, what you see on the left is actually consistent with what we had seen before. With the single-dose data is a robust and rapid reduction in pruritus, both in terms of the week 1 response that we reported as well as the week 12 response. Now, these are the aggregated data in all patients, but when you break it down and look at the patients who did not require topical corticosteroid rescue in the study, and that's in the open circles, you see essentially the true horsepower of the mechanism in reducing pruritus via inhibition of the OSMR beta axis. The closed circles represent, the closed triangles represent the aggregated effect of KPL-716 in the topical corticosteroids. Now, in terms of the responder analysis, the worst itch reduction of 4 points or greater is considered by many to be a clinically relevant or clinically meaningful outcome, and what you see here on the right-hand side are those patients with placebo, and you can see that those that responded in general tended to do so to very low levels, hovering around 4, whereas on the left-hand side, what you see here is the KPL-716 patients, as we had seen in the first single-dose study, tended to have very robust reductions in worst itch NRS, categorically, not only -4, but all of them 5, 6, 7, and 8, and even 9-point reductions in pruritus. Now, these data, in terms of the robust antipruritic effect, really then power us forward into the phase 2 program in prurigo nodularis. And as Sanj mentioned, this is a debilitating skin condition where there's a disruption of the neuronal architecture, which results in an itch-scratch cycle that's relentless. And these patients develop nodules in the skin, a terrible quality of life. At this point, there's currently no approved therapy for this disease. We are now in the middle of a phase 2A program, shown on this next slide. There we go. On the left-hand side, you can see the phase 2A portion in which patients are with prurigo nodularis and a high degree of pruritus are randomized 1-to-1 to KPL-716 or placebo for an 8-week treatment period. And the dose includes a 720 milligram subQ loading dose and the 360 milligram per week subcutaneous dose. And then, depending on those data, there is a phase 2B program as well. But of course, that depends on the data and the competitive landscape. In addition to prurigo nodularis, we are currently studying other chronic pruritic conditions in this, so to speak, basket study. You see the indications listed on the left. Similar dosing regimen, and this is more of an experimental medicine approach where what we're trying to do is link the presence or absence of the OSMR beta signature, so IL-31 and oncostatin M, with the presence or absence of treatment response to KPL-716 as measured by a reduction in pruritus. Both of these trials are now ongoing. The phase 2A study in prurigo nodularis, we're looking at data, we're expecting data in the first half of 2020. And with this study, we're expecting on a cohort-by-cohort basis for the data to roll out over the course of 2020. Moving forward to Rolana-cept. As Sanj mentioned, Rolana-cept is a once-weekly IL-1 alpha and IL-1 beta cytokine trap. It's already approved, and it was invented by Regeneron. It's marketed by them for CAPS, which is a rare genetic autoinflammatory condition. And right now we're currently enrolling the phase 3 trial Rhapsody for the treatment of recurrent pericarditis, which is again a debilitating cardiovascular condition of chest pain that's worse when lying down, relieved sometimes by sitting up, but importantly has other complications which can include a buildup of fluid around the heart, which can compress the heart, as well as just an overall really reduction in the quality of life of these patients. And at this point there are no approved therapies, as Sanj mentioned. Now, when we look at the treatment paradigm for these patients, which is on the next slide. There you go. Great. These patients are treated initially with NSAIDs, then colchicine, and then ultimately topical corticosteroids. And as patients develop refractory disease, they become more and more dependent upon these oral corticosteroids in order to maintain their disease. And thus, the morbidity of the disease is not only the disease itself, but also the treatments that are used in order to manage it. And so thus what we see are many of the complications of chronic corticosteroid use. And so as we think about the potential role of Rolana-cept and the way that we are studying it, we're studying it not only for the potential treatment and prevention of recurrent pericarditis, but also to explore the possibility of actually coming off of topical corticosteroids, maybe even obviating the need for steroids, thus a steroid-sparing agent. And that's how we've designed our clinical trials, and we have early data to support that. Now, in terms of working through the mechanism of what is it that we can do to make things better for patients in this disease, the data really point to a pivotal role of interleukin-1 alpha and interleukin-1 beta in this disease. Interleukin-1 alpha is a sentinel molecule that starts off inflammatory cascades when there's tissue injury, and that's then augmented by the role of the inflammasome and IL-1 beta production. And all of that results in a recursive vicious cycle that then causes perpetuation of inflammation and tissue injury. Now, we have established proof of concept in our open label phase 2 program. We have reported these data at the American College of Cardiology. And we studied different pericarditis patient populations, as seen here. We'll focus our attention primarily on part 1, which is the symptomatic patients who present with elevated pain, and elevated C-reactive protein is a marker of systemic inflammation activation. And these patients were treated with a marketed dose of Rolana-cept, which is a 320 milligram subcutaneous load, followed by 160 milligrams weekly maintenance, and there was a 6-week base treatment period. And then there was an additional 18-week treatment period and an optional extension where patients could wean off of their standard of care medications. At the time of the data cut that we showed at American College of Cardiology, we had, of course, about 26 patients enrolled in the entire study, 12 subjects who had completed the base treatment period, and some of whom were extending into the long-term extension. And what we saw in this data set is a reduction in patient-reported pain, as using an NRS scale, as well as C-reactive protein. And you see that the time to CRP normalization is in 9 days. And what you can see also is a persistent effect over the course of not only the treatment period, but also into the extension. Important statistics are listed for you here in the box. But in addition to these reported outcomes that are measured here, data that we have on our website also show resolution of pericardial signs and symptoms of inflammation, which really points to management of the totality of the disease and not just symptomatology. And then looking forward to this concept of potential steroid sparing, at the time of the data cut, there were 4 subjects who had entered the trial on topical corticosteroids, 5 subjects who had entered topical corticosteroids at baseline, and subjects, 2 of whom had completed 24 weeks of treatment. They were actually able to come off of their oral steroids, which is really a promising finding with regard to thinking about the potential for steroid sparing. In terms of the overall safety profile, well tolerated. The side effect profile is consistent with the Rolana-cept label. Most common adverse experiences are injection site reactions. There was one serious adverse experience of a skin abscess that was where the patient discontinued treatment, that was managed with antibiotics, and infections are, of course, reported in the label. Now, we're expecting final data from this trial later this year. Finally, as Sanj mentioned, we are now in a single pivotal phase 3 clinical trial, which is called Rhapsody. This is a randomized withdrawal study design, a global study, 50 subjects running in 4 countries. We are presenting the design of this study at the European Society of Cardiology meetings in Paris later this month. The data from this study are expected in the second half of 2020. So finally, moving on to Mavar-illuma. Mavar-illuma is a GM-CSF receptor inhibitor. Of course, this is an antibody that had been well studied by MedImmune through a phase 2b program in rheumatoid arthritis. It was successful and met all of its endpoints. We are now focusing Mavar-illuma on giant cell arteritis and other inflammatory cardiomyopathies. Now, giant cell arteritis is an inflammation of the muscular arteries of the body. Important complications can be blindness from interruption of the blood supply to the eye, and there's also a large vessel form of this disease which can cause aortic aneurysms. We're now enrolling, as Sanj mentioned, a phase 2 trial in giant cell arteritis. Now, in terms of the management of giant cell arteritis, in general, these patients are treated with oral corticosteroids. And what we see in the management of this disease, which tends to be a very long-term disease, often measured in years, is that as the steroids are tapered, what we see is the disease tends to come back. And so again, this becomes a situation of the morbidity of the disease combined with the morbidity of long-term chronic steroid use. Now, there is one additional therapy that has been recently approved as an adjunct to steroid taper, that is tocilizumab. Tocilizumab blocks IL-6, which is relatively downstream, actually works at the level of the liver, blocking the production of C-reactive protein. So while it is a relative advance for patients over oral corticosteroids, it still does not necessarily address all of the fundamental mechanisms of the disease. We're going to go into that mechanism in a moment, and therefore there remains a significant unmet medical need. Now, focusing forward on the mechanism of GM-CSF. Now, this is a bit of a busy slide that has a lot of references that you can read, but what it basically tells you is that GM-CSF is an important cytokine in driving the key pathway that leads to the pathophysiology of this disease. And the pathophysiology of this disease is driven by primarily interferon gamma and the formation of giant cells, so Th1 inflammation. Th17 inflammation also does play a particular role in this disease. And we've been doing work in this space, which up until now had not been particularly well characterized, really putting together all of the links in the chain, starting with GM-CSF and coming out on the other side, which is, of course, the pathophysiology of the patients. And so what we have here are some data that we presented at the EULAR meetings in Madrid, just earlier this summer. And what it does is it shows upregulation of the genetic signature, not only of GM-CSF, but also of the receptor, as well as transcription factors that are downstream of that, as well as other inflammatory markers in the arteries of disease patients compared to those of control patients. In addition, a particularly important finding is elevation of the Th1 signature in these biopsies. Now, this is important because many of these patients, we believe, were treated with oral steroids before their biopsy was obtained. And so what we're seeing is elevation of the interferon gamma and Th1 signature despite steroid therapy, and that may be, in fact, part of the reason why this disease is so difficult to manage. Now, moving forward to the actual results with Mavar-illuma, in a co-culture experiment with similar groups of arteries, what was seen is downregulation of the GM-CSF pathway as well as downstream mediators with Mavar-illuma versus placebo. Where we are now is, of course, enrolling a phase 2 clinical trial, which is also a global trial in 60 subjects with different types of giant cell arteritis, Mavar-illuma versus placebo, on top of a 26-week steroid taper. With that, I'll turn it over to Sanj.
Thank you, John. Thanks. So this is where we are today in terms of our programs. We're rapidly accelerating our pipeline programs. In terms of cash, our Q2 cash balance was $287 million. Importantly, we have no debt, and that means we're well capitalized to reach our milestones and fund the company into 2021. John mentioned we have a number of milestones. I'll just recap those briefly. The full final data set from the phase 2 Rolana-cept study this year, the phase 2A data from Patel and Mavar-illuma in the first half of next year, multiple data cohort readouts from the chronic pruritus studies throughout 2020, pivotal phase 3 results in the second half of next year, global phase 2 results from Mavar-illuma. We have got a lot going on, a lot of exciting news coming up. I'll close by saying thank you. 3 important comments I mentioned to David earlier on. Kiniksa likes to execute and meet our timelines, and so far we've delivered on all of them. Secondly, every second counts. And thirdly, I mentioned to him that Wedbush or Parker needs to invest in better clickers so we could advance our slides in time. But otherwise, thank you very much. Cheers.
Or one from me. So just to recap on the pruritus data that we talked about earlier, you press released earlier. Could you run through, again, the magnitude of benefit that you saw, and we have seen other, itch reduction strategies. I know it's tough cross-trial, but what do you think your advantages are if you think about other clinical trials and what the scores, what the data have seen to date?
Yeah. So in terms of the reduction in pruritus, when you look at the 2 slides that we have there that show the magnitude of response in terms of reduction of pruritus, these are very robust. They're very fast, so among the fastest in terms of the speed of response, and then you see the durability of that response over the 12-week treatment period. I think if you're looking for the data that show the maximum horsepower of the mechanism, that just because of the way the signals played out in the trial, the open circles in the graph on the right that you'll see in a second really show the kind of maximum effect of change versus baseline of about minus 60%. So in that sense, that reduction is very competitive. And I think what it does is it sets us up very nicely for the prurigo nodularis work, and especially in comparison to some of the other data that have been released. You're not able to go backwards. That's okay. So all of those data are on our website. But I think as we think about the phase 2A study, where we're taking in basically a proof of concept dose into the trial in order to measure that reduction in pruritus, the trial is well designed and well powered in order to show that effect. Oh, here we go. Yeah.
Remember that for advice. On the, have you disclosed the powering of the phase 2A?
I would disclose the powering.
Yeah.
So we rely upon the literature pretty much as everyone else has in terms of, for example, the expected placebo effect. Well, the effective placebo effect in terms of injection of placebo. We've actually completed our own work in this space, which is the LOTUS-PN study, which is a longitudinal observational study where we actually took 54 individuals and measured them over the course of time. And so we actually have some of our own internal data. So these data that we just showed from the repeated single-dose study do show, though, that the reduction in pruritus in terms of variability, et cetera, is actually very tight. So there's a very strong and tight response. And so I think with the 80 to 100 subjects that are in the study right now, that was the kind of initial powering. But as Sanj had actually mentioned, we are looking at potential ways to accelerate that depending on how the trial is playing out going forward.